Hannah Davis from Hayti High School, and I'm a senior. And in my project, I looked at identifying the differentiation pathway and cell type of the newly discovered dual receptor expressing lymphocytes in relation to type 1 diabetes. And to give a little bit of background information about my project, type 1 diabetes is an autoimmune disease in which the body attacks its own islets of insulin-producing beta cells within the pancreas. And diabetes is a disease of the immune system. The immune system is the body's first defense against intruders known as pathogens. Within the immune system, you're going to have lymphocytes that officiate immune response. However, when this goes wrong, autoimmunity occurs. And this occurs when the immune cells target their own functioning cells as faulty cells and destroy them. So one new cell found in relation to autoimmunity is the dual receptor expressing lymphocyte, or the DE cell. And this cell displays both a T cell receptor and a B cell receptor. And according to the article in which these cells were originally published, they are shown to mimic an insulin peptide to trigger diabetes-specific helper T cells. And in the article, it was stated that these cells have the potential to be the driving cause of type 1 diabetes, although this was not adequately supported by the article, and many scientists found that the actual function of this cell could not relate to type 1 diabetes. So in my project, I chose to interpret the biological data from the project in which the DE cells were originally published through bioinformatics. And within bioinformatics, I used RNA-seq analysis technology, and within that, I used differential gene expression technology. So why did I choose this project? First, in terms of societal impact, the DE cells are new. They were discovered under a year ago, and we need as much information on these cells as possible to see if they are important in learning about type 1 diabetes and potentially other diseases. Furthermore, identifying important information about these cells, such as the function and developmental pathway, if they are responsible for type 1 diabetes, could play a role in development of immunotherapies or prevention methods for type 1 diabetes. And in terms of personal connections, my family has a history of type 2 diabetes, and we are all predisposed to the disease. So I felt if I could connect this cell from type 1 diabetes to type 2 diabetes, I may be able to eliminate that genetic factor from my family's background. Furthermore, I have an interest in the field of bioinformatics and genetics in terms of a future career. So this is what I did in terms of a procedure. First, I downloaded the raw data from NCBI, and then I ran a pipeline on a bioinformatics platform provided to me from Science Coach, and I used a platform for differential gene expression analysis that output a normalized count in terms of FPKM, which I used to compare different gene expression output levels. And then I drew conclusions based on the levels of output. So this is the DE cells top 50 expressed genes. And within these top 50, some important genes I'd like to point out are these MT genes, which are going to dominate the majority of the top 50. This would indicate that there is going to be a high proliferation rate because these are mitochondrial RNA genes. Furthermore, the natural killer granzyme protein, NKG7, being in the top 50, also indicates that this cell has a cytotoxic nature. This is the natural killer gene signature being presented by the DE cell as well. And within this, I'd like to point out the expression at very high rates of perforin, as well as a granzyme being GNLY. This is the B cell gene signature being expressed at a high rate by both the DE cell and a B cell in the B cell light, which indicates that the DE cell is going to have that activated BCR on its cell surface. So in conclusion, in relation to the cell type, the DE cell is going to possess the majority of lineage markers indicating that it's a CD8 or cytotoxic natural killer T cell, which is a natural killer cell with a TCR equipped, which suggests that the DE cell is expressing B cell genes because of a germline encoded BCR. This is the proposed differentiation pathway. You're going to see that the DE cell is going to follow a relatively standard differentiation pathway, but it's going to stop at the natural killer T cell stage, and its TCR is also going to be activated by self-antigen binding, which gives a reason for its autoimmune qualities. This would be the proposed cell function for the DE cell. So the DE cell is going to either identify the beta cell islet through self-antigen identification because of those autoimmune properties, or it's going to mimic insulin like stated in the original article, but either way, this is going to trigger cytokine excretion in the form of Th1 and Th2 signaling to the TCR, which will trigger cytotoxic activation in the form of perforin and granzymes, which will attach to the beta cell islet and trigger apoptosis. So what does this all mean? Well, a cell of this type and function has never been explicitly identified. A natural killer T cell with B cell features, or a cell that is cytotoxic with antigen identification features, indicates that there's a much closer connection between the innate and adaptive immune system than previously described in immunology. This also means that the developmental pathway of immune cells is not absolute, and that many cells within the immune system could have features of both cytotoxic behavior and antigen identifying behavior, which means that this mutant lymphocyte and many other mutant lymphocytes could be going under the radar of researchers because of their unique properties and having dual expressing properties. And so this cell can contribute perhaps to other autoimmune diseases and even cancers. So the future of many diseases and type 1 diabetes relies on more research being presented regarding these cells, excuse me. Do you have any questions?
Anybody is able to ask questions in the group chat, and I do see some questions coming in. Why did you had mentioned that you had a family background of the type 1 diabetes, type 2. So how do you plan on going forward with this? What do you want to do in the future?
Well, the first thing I'd like to do is actually studying these cells in person, since I was only using biological data. And by studying these cells in person, I would be able to hopefully confirm these different cell markers that I found through the gene expression, and hopefully identifying this unique genotype in other diseases could lead to it being implicated in other cellular pathways.
So a question coming in, I'm not sure how you can answer it, but maybe you can. So when you are dealing, what do you hope that this dual expressor cell is going to show us in regards to the type 1 diabetes? I guess it means how are you actually going to use this information?
Okay. Well, with the dual receptor expressing cell, the cellular pathway of type 1 diabetes, many researchers are unsure why the body is actually attacking its own beta cell islets and triggering that apoptosis. So this mutant cell that's gaining its autoimmune qualities from this natural killer cell background, which means that it's not going to recognize self, means that this cell could potentially be reprogrammed in order to be recognized as self. So hopefully if I can support enough that this cell is actually causing the disease, the number one driving factor of this disease, I can potentially go in and try to reprogram the cell and see if that would help in reverting the effects of the disease.